Hello students, this is Dr. Faizan Mirza. I'll be discussing question 21 till 40 in this video of Feb March 2022. This is MCQ paper. So I already have covered the first half of the paper in the other video. So I'll be continuing this from question number 21. Question 21 is about some parts of typical human chromosome are more numerous than the others. Again, this is something of fact. You just know that histone proteins are more than telomeres and DNA molecule, because around every DNA molecule, one DNA molecule will, for example, if this is one DNA molecule, there are bases on this one DNA molecule. You will have telomeres at its top and its bottom. So one DNA molecule will have two telomeres. If it's having two telomeres, how many histone protein will have? It will have a lot of histone proteins all along it, because the histone proteins allow the DNA to get wrapped and to get condensed. So histones are most numerous, followed by telomere and the DNA molecule. Question 22, which row correctly describe the stage of mitosis? So if you go to the stage of mitosis, prophase, nuclear envelope disappears. Yes. Centromeres replicate. No, the centromeres do not replicate in prophase. The centromeres uh, the centromeres do not replicate in mitosis. You can just recall that. So and in fact, centromeres, when do the centromeres replicate? Centromere represent in the chromosome in this way. So do the centromeres replicate? No, they just do not replicate. They just split at the end of metaphase. So metaphase, nuclear envelope not present. Yes, they move to the opposite poles. Centromeres move to the opposite poles. No, at metaphase, the centromere splits from the center. So the centromere splits at metaphase at the end of mitosis. So at the end of metaphase, not at the end of mitosis, at the end of metaphase, the centromere splits. So the option B is incorrect. Anaphase begins to reform. Nuclear envelope begins to reform. No, that's not correct. So you have to logically think for every option why each option is wrong. So this again needs working. So this question will take obviously more than a minute or two. So stage of mitosis, telophase, yes, nuclear envelope reforms. The centromeres will be pulled apart because it was separated at the end of metaphase, and the spindle fibers they start to break down. So D is the answer. The diagram represents nucleotide containing adenine. They have given this diagram. The student may think this is ATP. This is not ATP. They are saying the diagram represents a nucleotide containing adenine. So students can go for the option that this is ATP, because somehow it looks similar, although it's not at all ATP. Keep that in mind, because the ATP will have three phosphates here. So this is not an ATP. Coming back, which statements about this nucleotide are correct? We know there's a phosphate group, there's a nitrogen, this is the pentose sugar present, and then there's a base which is adenine. We know adenine can bond with thymine or uracil. Let's move on. The carbohydrates pentose, the base is nitrogen. A container at the base pairing occurs with uracil. Adenine is a pyrimidine base. No, it's a purine base. So 1, 2, 3 are correct. 4 is wrong. Which row describes which row is correct for the replication of DNA molecule? We know that the DNA molecule replicates. The DNA direction of synthesis is always 5' to 3'. So option C and D can be correct. But which enzyme catalyzes this? The DNA polymerase is the enzyme that catalyzes the reaction in the direction of 5' to 3'. DNA polymerase add nucleotides in the growing chain in the direction of 5' to 3', while the template is runs on 3' to 5'. So if this is the template strand having 3' end here and a 5' at the opposite, the DNA being the newly synthesized strand will go 5' to 3' in this direction, in this way. So that's how it will keep on going. So it will be going in this direction. 5' to 3'. DNA ligase is joining phosphodiester bond. The DNA polymerase is the enzyme that aligns that keeps on replicating the DNA strand and allowing the complementary base pair to occur. Question 25. The students were asked to look at a photomicrograph of a cross section of an unfamiliar material. Describe what they could see. So they could see circles. You can see these are circles of cells. These are cell F. The student described the cross section of F as being circular. Yes, a hollow tube and spherical. Which descriptions are correct? For F correctly stated, the student could actually see. What can be seen? Can they see it circular? Yes, the student can see. Do they see a hollow tube? No, the diagram doesn't show to be a hollow tube. This is not visible in the diagram. Similarly, is it a spherical structure? No, the diagram is not a 3D diagram. There's a difference between circle and sphere. A sphere is 3D. A circle is on 2-dimensional plane. So yes, these are circles. So option only 1 is correct. Kind of a strange question though. Question 26, but it was checking your observation skills here. Question 26, the diagram shows the relationship between phloem C tube elements and xylem vessel elements and companion cells. So you will just keep on seeing which structures can be lined up accordingly. So 2 must be common between 1 and 3. So if 2 is endoplasmic reticulum, is it present in companion cell? Yes. Is it present in phloem? Yes, it's present. So it's common in both. Now 4 must be common between 3 and 5. So 3 is a phloem C element and xylem vessel elements. They both should have something in common. And what's in common? They both lack nucleus. Why does an air bubble in xylem vessel element stop the flow of water? We know that loss of addition, loss of cohesion, collapse of xylem vessel. No, xylem vessel cannot collapse because of what the air bubble. Xylem vessel is very strong because of lignin, so that's not going to happen. So 3 is incorrect. Now coming to 1 and 2, why do we choose cohesion or addition? We do not choose which one will be impeded because of an air bubble. Addition, we know water will form bond with the vessel, and with that it can still cross plasmodesmata, it can cross pit, it can go to the surrounding xylem, which is still working. But if there is a loss of cohesion, the water molecules with each other will not bond because of an air bubble present between them. So what's actually happening here? The answer will be 2. Why? Because why does an air bubble in xylem vessel element stop the water flow? It stops the water flow because it affects the cohesion. Water molecules cannot cannot pull each other anymore, cannot be attracted to one another, because between every water molecule or between a few water molecules there might be an air bubble and cohesion will not be occurring. But the water molecule can still be forming bonds with the vessel wall, and it can still move on from one xylem to another through the plasmodesmata or through the pits. 28. Which changes occur in the water potential and the volume of solution the phloem C tube occur when amino acids are moved into sink from the phloem C tube? We know that during unloading at sink, sucrose move out into the sink, water potential of the C tube element increase, and solution of the solution volume start to decrease. So our option A is correct because water potential in the phloem C tube becomes higher, volume of solution phloem C tube decreases. Which feature of some xerophytic leaves reduce the rate of transpiration by decreasing the water potential gradient between the internal leaf structures and the atmosphere? We know that these xerophytes have sunken stomata. The sunken stomata are present in the crypts. Now, when these sunken stomata are present in the crypts, this region is not having external air. External air is present outside in the outer atmosphere. So is the internal part of your leaf in contact with the external air? No, it's not in contact with the external air. It is in contact with this air present in this moist region. As a result, the gradient will not be very sharp. If the stomata was in direct contact with the external environment here, in that case, it would be a very steep gradient. There is moisture here, there is moisture here, inside as well, the gradient is less sharp. So stomata located sunken pits on the leaf surface, they prevent the leaf from not being exposed directly. A thick pexicuticle on the upper epidermis, no, we do not know if the stomata are on the upper surface only. These reduce to spines, they reduce surface area to volume ratio. But how would this prevent the water potential gradient between the atmosphere and the leaf surface? There is no relationship. Elongated leaves are swollen, storing large amounts of water. Again, this is not the case because you have to link it with the potential gradient of water. Now the diagram shows a cross section of a mammalian heart. This is a tricky question because this time they just flipped the diagram. Normally we don't have the left side being visible on the left side. So in biology we always have diagrams in which the diagrams are flipped because this is exactly how you look at an image in a subject sitting next to you or a body that you are studying. But this way they just reverted this diagram to confuse you. Now you have to make sure that you keep in mind that this is a cross section through a mammalian heart. That's what they have stated. These are the chambers H and G. How do you identify which chamber is what? You just look at the vessel, the wall that surrounds this chamber. If the wall is thick, then definitely that's a left ventricle, irrespective of whatever shape, whatever side it might be. Similarly, if the wall is thin, this must be the right ventricle. So you identify left ventricle as H and G as the right ventricle. Now, when you look for the option, you see that they already mentioned H here, but they flipped H on this side of the column. So if you were looking at left ventricle here, you would have made A as a correct option, which is wrong. So they made you realize that, okay, H is left ventricle, but now they want to trick you again. So they placed H in this column and G, which is right ventricle, it goes into the opposite column. Technically speaking, G should have been here, H should have been here, but now since they are giving you 1 hour 15 minutes, they will trick you. These are the tricks that the examiners are playing. So you need to keep in mind that you look at the options very well. If it's H, you have to look for the column of H and not for the column of G. So right ventricle and left ventricle, we know G is the right ventricle and H is the left ventricle. We move on to the next question. 31, blood entering from vena cava well passed through or passed or several of these structures before entering the lungs. 5 of these structures are included in the list. We just make our own list. Vena cava from the blood from vena cava goes to right atrium, then the sinoatrial node, then the AV node, then it goes to the tricuspid valve, which is AV valve. It will go to the right ventricle. It will pass through the Purkinje fibers. It will move to the seminal valve. It will go to the pulmonary artery and it will go to the lungs. Now, of all the structures mentioned here, if we see sinoatrial node will come first, I made it 1. AV node will come second, Purkinje tissue will come third, seminal will come fourth, pulmonary artery will come 5. I just labeled these accordingly here. Again, working is required. You must have a pencil in your hand, so you keep on annotating accordingly. Now, after arranging these 5 structures in the correct order of blood flow from vena cava to the lung, which structure will be third? So which structure will be third? We know Purkinje tissue will be third. So you need to make sure you recall what you have studied and be able to align this appropriately. The bar chart shows the quantity of endothelial cell, elastic tissue, and muscle tissue in 3 blood vessels, P, Q, and R. You need to identify these. Which of these is which? So we know in vein is, how do you identify that R is vein? Because vein will have least muscle tissue. So if we look at the muscle tissue bar, it should be the shortest one. So this is the only vessel in which it's shortest. So R should be vein. So the option could either be A or it could be C. But then we come to elastic and muscular arteries. We know that elastic artery should have more of the elastic tissue. So this is elastic tissue more. This elastic tissue is less, more of the muscle layer here. So we know that P is elastic, Q is muscular, R is vein. So option becomes A. The maximum pressure in the circular force chamber of healthy human heart was recorded during one cardiac cycle. The maximum pressure recorded was 3, 10, 25, and 120. Which value is recorded for the right ventricle? We know that right atria will have the least pressure of the entire cardiovascular system. That's about just 3 millimeter mercury. That's a fact. And the highest pressure is between the left ventricle, which is 120 millimeter mercury, and coming off to the ventricles. The out of these, which one will have which one will be left ventricle, the left atrium, and which one will be right ventricle? Right ventricle is a ventricle after all. It should have a pressure higher than the atria. So 25 millimeter mercury sounds more logical. The atria should have the lesser pressure. Ventricles should have a higher pressure. So our option becomes 25. The graph shows pressure changes in the three structure on the right side of the heart. Now, instead of giving the left side, they have given the right side. So again, the same kind of valves are working. You just need to recall which valves are opening and closing in which direction. We know this is you just label. This is the right ventricle curve. This is right atria, and this is the pulmonary artery. What is represented by the line table labeled T? So T is actually representing pressure in the pulmonary artery, because on the left side, this line represents the pressure changes in the aorta. Which statement helps explain why there is no cartilage in the walls of bronchioles? Cartilage would make bronchioles too narrow, not linked with lumen diameter. No, this is just not linked with lumen diameter at all. Gases must diffuse. No, bronchioles do not allow any gas exchange. Smooth muscle is sufficient to provide support to the walls of bronchioles. Yes, this is correct. The bronchial wall do not need to change shape. They need to change shape. This is how a plan diagram of, or not the plan diagram. This is having a lot of breaks here. This is how the tissues are supposed to be in a bronchiole. There will be a lot of smooth muscles and elastic fibers around in a bronchiole, and the lumen will have endothelial lining or epithelium which is folded. It will always be folded so that when it enlarges, it becomes unfolded to accommodate more air going through it. So it can allow change in shape. This can only occur if there's contraction, relaxation, or smooth muscles present around the bronchiole. So yes, C is correct. Again, this is quite strange that they gave two questions for the same part of the gas exchange system. They asked bronchioles in the 35th question and 36 as well. Probably they were just running short of some topics from which they wanted to ask question. This is kind of strange. This never happens. This happens. This is a rare case though. The photomicrograph shows a cross section of a part of a gaseous system of a mammal. Again, they are showing you bronchioles. Actually, the examiner again trying to trick you that this is not the bronchiole. If you chose bronchiole, then this is definitely not a bronchiole. But this is a bronchiole. This is a bronchiole. These structures here outside are the alveoli. This is the gas exchange membrane, and this is how the gas exchange occurs at the gas exchange membrane. So you can just recall. Now from these folds, we'll just recognize that this is a bronchiole, and these are the smooth muscles and the elastic fibers. of this structure. So there, what is shown by the line I, diameter of alveolus? No, this is alveoli. If the line was drawn in this part, this would have been the alveolar diameter. The diameter of bronchiole, yes. The diameter of a capillary, no, capillary is not shown here. The diameter of trachea should have an incomplete ring of cartilage present around it, and it should have a very thick cartilage shown, in fact, a very broad cartilage. There's just no cartilage here, so it can't be trachea. 37. Which feature of a disease cholera decreases the spread of pathogen Vibrio cholerae? You have to just recall the prevention of Vibrio cholera. Immunity to cholera vaccination is short-lived, lasting less than 2 years after vaccination of 50% people. This will not decrease. This will not decrease the spread of infection. There's just no logic here. Up to 98% of people infected with Vibrio cholerae are symptomless carriers. No way. They are not symptomless carriers. There are no symptomless carriers of cholera. People suffer from diarrhea there. Cholera rapidly kills up to 50% of people's symptoms are not treated. Yes, any disease which is extremely fatal, any disease that kills a person in a very short time will have a reduced chance of spreading, because the person will die very quickly before the person could interact with others and transmit the pathogen to the other people. So any disease in that sort. So if your patient survives very long for a disease, for example, in TB, it's a slow infection. So the patients, they keep on surviving very long. They survive for various years, and they keep on spreading the disease to many people, many other people every time they come in contact with others. Simple rehydration therapy successfully 99%. No antibiotics are required. Yes, in fact, no, this is wrong. Antibiotics are required. I didn't mean to say no antibiotics are required. So, question 38. Which row shows how penicillin kills the bacteria? The process inhibition of penicillin, its formation of peptidoglycan crosslinks is prevented. This is a fact. Effect on bacteria, water enters and bursts this up. Breakdown of peptidoglycan crosslink? No, it doesn't cross breakdown. Formation of peptidoglycan, like a peptidoglycan crossing, correct. Water leaves the bacteria and dehydrate. There is no reason for the bacteria become dehydrated and dying. The bacteria just cannot die that way, na? So because it's a cell wall, the water keeps on entering, the cell wall becomes loosened and the cell bursts. In hybridoma, the breakdown is just not correct. So you have to choose between A and C. So A is the correct answer. In hybridoma method, what is grown by cell culture to produce monoclonal antibodies? Antigens, no. Lymphocytes, no. Myeloma cells, no. Clones, yes. So they have changed the term. They are just asking clones. Okay. What type of immunity does a baby have at birth? Active, artificial? No. It's neither active nor artificial. Active, natural. It is not active. Yes, it is natural. Passive, artificial. It is passive but not artificial. Passive, natural. Yes, it is immunity passed through placenta. So that's passive, natural. So that's it from my side. I hope you will be doing very well in your MCQ paper. I hope that was helpful for you. That's it from my side. Thank you so much.